We have learned more about the brain in the last 10 years than we have in all of human history. And it's accelerating every year with new techniques that become available. New discoveries are made. So when I was your age going to high school, I was told that you only use 10% of your brain. And I was also told that all the neurons that you have are present at birth, and it's all downhill from there. So we've learned a lot because Rusty Gage told us in the first lecture in this series that in the adult, new neurons are being born every day in a part of the brain called the hippocampus, where we think long-term memories are stored. So making more room perhaps for new memories. And as far as that 10% figure goes, I have never been able to track it down, but the 10% figure is probably based on wishful thinking, because we don't really know how much of our brain we actually use because of the fact that there's so much of it. In fact, there are 100 billion neurons in our brain. It's an astronomical number. 100 billion. How big is 100 billion? Well, if I had $1 for every neuron in your brain, I would be a wealthy man. But I couldn't cover this year's national budget deficit. Well, the first lecture was about new neurons being born in the hippocampus, and then in the second lecture, Nick Spitzer told us about how the neurons develop and interact, create networks, and then eventually allow us to be able to see. And we heard in the last lecture about especially the perceptual system of taste. How is it that it gets organized so that we can taste salty and sour, and the fantastic genetic techniques that are now available for being able to manipulate the genes that are responsible for perception. But in this lecture, I want to turn to the human brain. I want to turn to your brain. And I brought along a prop in order to show you what it looks like, because you probably have never seen your brain, certainly, but anyone's brain for that matter. But if you're a medical student, in fact, this is one of the rites of passage. They take you into one of the classrooms, and then you dissect a cadaver, and in particular, you take a look and see what's in the head. So that's what we're going to do here today. So this, in fact, I brought this prop with me from the medical school. And I've already done the dissection, so all I have to do is to take off the top of the head, something that you shouldn't do when you go home tonight. And lo and behold, look what we have in it. We have the top, the cerebrum, the surface that we can see from the very top, which contains all of our memories, our history, our hopes, our fears. They're all contained in this roughly 5-pound mass of cells. And we're going to now take it out so we can look at it and dissect it. And we can just hold it and look at it from the bottom and look at it from above. In fact, we can even take it apart, because this is a particularly well-constructed brain that has, as you can see, two halves, the two hemispheres, the right and left hemisphere, which in humans has become specialized. Language in most of you is in your left hemisphere, and spatial reasoning is in the right hemisphere. It's not perfect, but it's lateralization of function. So this reminds me of a story that was told to me by Maxwell Cowan. Max Cowan was here at the Salk Institute and was the scientific director before he moved to the Howard Hughes Medical Institute, which funds my research. And before that, before coming to the Salk, he was the dean of medicine at Washington University in St. Louis. And he told me the following story. He was asked to give a congressional testimony and decided that since it's very unlikely that any of the congressmen had actually seen the brain, that he'd actually take one with him. So he went down to the medical training area of the university, and being the dean, he was able to, of course, take one of the jars in which the brains were kept in formalin and put it in his briefcase and took it on the airplane with him. Back then, they didn't check your bags as carefully. And showed up at Washington. And there was a security guard in front of the testimony room. And the security guard asked him to open his briefcase. And so Max did. He opened his briefcase. And the security guard stiffened up and pointed at the jar and said, "What's that?" And Max Cowan said, "That's a brain." And the security guard said, "Why did you bring it?" And he said, "It's a backup. It's a spare brain. I always bring it with me just in case." And the guard, without hesitation, said, "Oh, you won't need it in there." So that actually raises an interesting question. Why do we have a brain, right? Why is it that nature gave us the brain that we have? Now, you don't actually need a brain to be alive, because there are plants that we know that some trees have lived for hundreds of years, right? And they're alive. They don't have brains, at least not the kind of brains that we have. They have cells, they have almost all of the basic ingredients, the basic elements of cells is the same in our brain as it is in those plant cells. They have membranes, they have proteins, they have actin, cytoskeleton, they have channels in the membrane, all of the basic housekeeping components. They make ATP for energy, and they respirate, and they have specialized things like chlorophyll and chloroplasts to convert sunlight. That's something that we don't have. But nonetheless, they're quite happy to sit there their entire life and just vegetate. But we have a brain in order to be able to move around, because once you have the potential of moving around, then you have the potential for finding new food resources, new places to go to find ways to nourish yourself. And after a long process of evolution, you can become a homeotherm. You can maintain a high temperature, which makes you independent, more independent of your environment. And that's really a large part of what evolution is all about, is overcoming the uncertainty of the world, trying to make sure that there's going to be a meal tomorrow, trying to maintain equilibrium. And the person who actually had that insight was a Frenchman back in the 19th century, Claude Bernard. And he was later, this idea of reaching an equilibrium and being able to maintain balance is now called homeostasis. And the basic computational principle is captured in this complex device called the thermostat, that most of you are familiar with if you've ever set the temperature in your house. How does it work? Well, there's a little dial, right, which is located right here, and you set the temperature that you want your house to be at, and the furnace fires up if its temperature is too low, and it will stop when it reaches the desired temperature, and it will maintain that, right? Maintains an equilibrium. And that's exactly the same principle that is at work in your body, maintaining your body temperature. But the brain has much more complex ways of being able to maintain your body through your ability to see and to navigate and to forage in the world, to find where to go in order to be able to maintain your energy sources. Because without the energy source, you won't last very long, and certainly you won't continue the species. Now, Theodosius Sejnowski, an evolutionist, once said that nothing in biology makes any sense except in the light of evolution. So we need to really ask the question of how it is that we actually got here. Where did we start? So the universe, astronomers tell us, is around 12 billion years old, plus or minus 3 billion years. It's hard to tell for exact, because nobody was around back then. But based on what we see in the heavens, the stars, the sun, the galaxies, we think that the Earth was formed probably about 4.5 billion years ago. And life started not too far after that, about 3 billion years ago. It was unicellular life. These were bacteria. And we know it was the origin of life because the bacteria and all life follows the same basic principles of being based on DNA. The structure of DNA was only discovered about 50 years ago by Jim Watson and Francis Crick, who is a colleague of mine here at the Salk Institute, who died just last year. And that was only 50 years ago that we made that discovery, right? Life has been going on for billions and billions of years. And we can follow the emergence of different species at different eras. And in order to illustrate this, I'm going to create a little timeline here. Okay, I'm going to put the little top hat here over in one corner. And I'm going to stand around, I've already paced this off, around 30 feet away, and pretend that I'm the Big Bang, right? That was the origin of the universe, 12 billion years ago, and that's a timeline. Here it's in a spiral. I'm going to make it a straight line just to illustrate the point a little more clearly. And now I'm going to walk, and this is the time during which the universe evolved, and now the stars are forming, the galaxies are forming and condensing, and finally our sun begins. This was now 4.5 billion years ago, right around here, right, somewhere in the middle. And then, about a third of the way to the present, life began. I'm a bacteria here. That's where it began. So we've come a long way, right? But it's not that much farther to go. So one of the major events that occurred probably in this region here, roughly 1 billion to 600 million years ago, was the emergence of multicellular organisms, and in particular eukaryotes. And that was around 600 million years ago, and according to my calculation here, that would be 3 feet, roughly 10%. So it's about 3 feet away. That's where the early invertebrates emerged that populated the oceans. Now, you probably are familiar with the fact that there are dinosaurs on the Earth, and they disappeared around 65 million years ago, right? 65 million years ago. So when did that happen relative to our timeline? 4 inches. Okay? 4 inches away. Dinosaurs are gone. Humans aren't here yet. In fact, hominids didn't appear on Earth until around 3 million years ago. The first fossil evidence that we have for creatures that stood up and looked something like us with big brains, that occurred around 1/4 of an inch from the present, right? I'm almost on top of this, 1/4 of an inch. Okay, what's left? Well, what's left is all of human history. And we've got to fit it into the last 1/4 inch. So let's think about language and humans. In other words, how long do we think we've been talking to each other? Now, there's no real record. We don't really know exactly, but it's thought that it's not more than a few 100,000 years, right? So a few 100,000 years is now, we're talking about a very, very small fraction of an inch, on the order of maybe 1/100th of an inch. Okay? And what about recorded history? What about written history? Writing is a very recent invention. It's literally an invention because it wasn't something that was discovered by biology. It was something that we created out of our own brains. We discovered a way to hand knowledge down from one generation to the next by coming up with written words that represented our knowledge and passing that on, adding to it using the scientific method. And that only happened about 15,000 years ago. The historical record doesn't go back much farther than about 15,000 years ago. And now you're talking about roughly, a thousandth of an inch. And finally, if you want to talk about, really understanding how, for example, the great discoveries in physics 100 years ago, the great discoveries in biology in the last 50 years, that's all in the last millionth of an inch. What's happened over the last couple hundred years is an explosion on the time scale of the universe. And even on the time scale of the human race, millions of years, it's also a very tiny fraction. So you have to understand how special a time this is in the history of the world. The fact that we're here today and you're listening to me and we have made all of these discoveries. It's really an amazingly rare event in the life of the universe. Okay, so the story that I want to tell you now in the context of evolution, because of the fact that evolution is very conservative. It really likes to use the same parts, the same principles over and over again. And the key to understanding the theme of today's lecture, which is brains, rewards, and addiction, is going to come from a very unlikely source. It's going to come from studies that were done around 100 years ago by a neuroethologist, by animal behavior specialist Carl von Frisch. So von Frisch studied bees, and he discovered a couple of really important features of their behavior. He discovered, for example, that bees had color vision. He discovered that bees could learn to identify flowers that are likely to have good food. And for a bee, that means nectar. And nectar is a nice high energy source of sugar, of fructose. And here is a bee sampling the nectar in the flower, and while it's doing that, of course, the pollen is getting on its legs and it's going around and pollinating other flowers. This is nature's way of synergistically making sure that life continues. But let's think about this bee. This is really an amazing creature. This bee, this particular worker bee, left its hive a couple of hours ago, traveled 3 kilometers. Now, you have to think about the scale of the bee. 3 kilometers is relative to the bee length, right? The bee is less than, say, a centimeter. And so it's on the order of, let's see, a kilometer is 1,000 meters, and 100,000 centimeters. So 100,000 of your body lengths, which is, say, 10 feet, is 1,000,000 feet, right? We're talking about going up to Los Angeles. It's like you're going foraging for some food up in Los Angeles. They have to fly out. They have to figure out where to go. So they have to navigate. They have to keep track of landmarks. And they get there. And they go onto a nice little flower here. You notice this flower here has a particular shape, and it has a particular color, and it has a particular floral essence, a certain olfactory smell. The bee can remember all 3 of those things. It can remember the color, the form, and the smell. And not only that, but it can go back to the hive, all the way back, from LA back to San Diego. It can tell its fellow bees by wiggling its behind, something that Carl von Frisch discovered called the bee dance. And it can tell the rest of the worker bees where this great source of nectar was, so that the rest of the bees can go and get it too. That's an amazing skill that these bees have. They're champion learners. Amongst the invertebrates, they have the best memories, amongst the best memories. And here's an experiment that he did. What he did was to feed these bees on some sugar water that was sitting on top of a blue plaque. And then the bees came back the next day. There was no sugar anywhere, no sugar water. And the question is, which one does it go to? Do the bees go to the blue one, or do they go to the other levels of gray? Well, it turns out that they preferentially, as you can see, go back to the blue one, even though there's nothing in it. And that's one trial learning. They can learn with 80% accuracy in just one shot. That's pretty good. That's better than a lot of the students in my classes. But of course, their livelihood depends on this. If they don't get this right, they're going to send a lot of other bees off in the wrong direction and to the wrong place, in the wrong patch. So this is really important for their survival. Evolution has put a lot of effort into making sure they can do that really, really well. And how do they do it? Well, we've got to go into the bee brain to figure this out. And that was done just very recently by a postdoc in the lab of Randolph Menzel, who is a neuroethologist in Berlin, who studies bee brains by putting electrodes into their tiny brains and recording from single neurons and asking how those neurons respond to different kinds of sensory stimuli, and in particular stimuli that are important for learning how to associate, say, an odor with the reward, the sugar reward that the bee is given. So here's Martin, and here is his discovery. It's a particular neuron in the bee brain called VMX1. You don't have to know what that means. It's ventral unpaired medial neuron, MX1. Here's the cell body down here. This is a cross-section of the bee brain. The whole thing is about 1 millimeter across. This is really ultra-compact. And what you're seeing here is the output of that neuron, namely the axonal arborization. This neuron, when it fires a spike, an all or none event, it releases a chemical at these little endings here, these little terminals that are called synapses. And you'll notice that there are regions of concentration. Well, this is the area where we know there are neurons that get input from the antennae, where there are sensory olfactory receptors. So this is called the antennal lobe. This region here is a very interesting part of the bee brain, which also gets, as you see, a very heavily innervated projection, axonal projection. This is called the mushroom bodies. You don't actually see anything in it because it's all outlined, but there's chock full of very, very small little neurons. And it's thought that this is a part of the bee brain that's really important for long-term memory. A lot of work's been done on that very recently. And finally, we see that this neuron also projects to these little lateral regions here, which are part of the bee brain that controls the proboscis. So it's a motor structure. So isn't it interesting that this one neuron, it sends messages to a sensory structure, a memory structure, and a motor structure. Well, what kind of message is it sending? So Martin recorded from this neuron, and here is a recording. These little upward strokes here is a single action potential. And the time base here is quite slow. This is 2 seconds. So this whole recording here lasts for about maybe 12 or 15 seconds. Now, at this point here, he put a little drop of sugar water on the proboscis of the bee and elicited a very high density, a very high firing rate of these spikes. And that's shown here. This little raster here shows the density of spikes. And then it continues on for at least 4 or 5 seconds. So that's interesting. So this, in a sense, is an internal measure of the degree of reward. If you put on a higher concentration of sugar, you get more spikes, less concentration, less spikes. And this message of the degree to which the reward is, has a high value or low value is being transmitted to those sensory structures. And now the question is, how can the bee brain use that information in order to be able to guide its future behavior? Because the key is trying to make a prediction about the future. You want to predict, if I land on this flower, what's the probability that it's going to be a rewarding experience? If I decide to go to a different flower, will I get more nectar or less nectar? I have to make those decisions on the basis of my previous experience, and I have to keep track of that. How is that done? Well, it's done with a neuromodulator called octopamine. Octopamine is a substance that's released by VMX1 onto the neurons in those different regions of the bee brain, and it changes their firing output in a very specific way. Around 10 years ago, two postdocs in my lab, Reed Montague, who's now at Baylor, and Peter Dayan, who is now the director of the Gatsby Computational Neuroscience Unit at University College London, developed a very simple computational model to explain all the bee data that I've just been telling you about. And I don't have time to go into the details, except I'm just going to give you the essence of the model. The essence of the model is this little circle here called P, and P stands for prediction. And in the bee brain, we think this is the octopamine neuron, VMX1. So this neuron, we know from the data I showed you earlier, gets an input, direct input from the nectar. You can think of that as the internal reward pathway. That's giving the bee the signal about how good it was, that last flower that you happen to land on. Now we know that these neurons, that this VMX1 also gets sensory input. And in particular, it gets visual input and it also gets olfactory input. So we know that there are direct projections coming from the sensory input. Now, before the bee does any learning, these sensory inputs have very little, or if any, drive to the neuron. They're neutral. They don't produce very much output. If you give the neuron, for example, if you give the bee blue sensory stimulus, the bee will ignore it because it doesn't particularly like blue. However, if you pair blue and a little bit after the blue, the nectar reward, then the next time that you give blue, you will drive that neuron. And that's because the strengths of the connections between the blue sensory input and that neuron has been strengthened. And it's been strengthened very quickly in a single trial. And what Reed and Peter did in my lab was develop a very simple algorithm, a computer model, for how that weight should change according to the relative timing of these two inputs, the sensory input and the nectar input. And now, if you think about this for a second, this is actually a very useful neuron to have. Why? The reason is that once you've trained up the weights, figured out which features, sensory features of the flower predict reward, you can now use that for being able to navigate by telling the motor system, for example, to land on the blue flower, right? And that's why we think that there's projections to the motor system as well as to the other parts of the bee brain. But the key thing that came out of this model, which is called the temporal difference model, is that the output of this neuron is making a prediction. It's making a prediction about whether this sensory stimulus that I'm getting right now is going to lead to a good event. Is it going to help me forage? Is it going to help me find a good patch of flowers? Now, what if I think this is a good patch? Suppose it's blue, and I land on it, and there's nothing there. And this happens, during spring when flowers bloom, and then they, of course, disappear, and now the bee is disappointed. Well, if the P neuron is firing at a high rate because it expects reward from the blue, but nothing comes in, then according to this algorithm, you should decrease the strength of that weight. You should lower your expectations. And that way, you can use your previous experience to online constantly readjust your expectations for what the likelihood is in the world of being able to get rewards from one place or another place, and to be able to weigh those options so that if you have a choice between yellow and blue, you can say, well, which one is better? I can sort of, by comparing the output of the P neuron, if P is firing more for blue than yellow, then I could bias my landing on the blue flowers. This sounds like a very simple kind of learning. It's called classical conditioning, but it turns out that mathematically we proved that not only is this an effective way of learning, but in principle, you could use this algorithm in order to learn a sequence of actions that will take you to a reward in the distant future. By back-chaining from the reward, you could learn, for example, that a particular patch of flowers flying over it is going to be a good place to go to from a distance because of the fact that you had been rewarded by blue in the past, and there's a blue patch down there. And you can back-chain that all the way to the hive. So in fact, this is a powerful way of being able to go through the world and learn sequences of actions, because you may not get rewarded the first time that you do something. You may have to go through a whole process. And an example of that would be for you students, for example, taking tests, right? You take a test, you study for the exam, and your reward is going to be the grade you're going to get way in the future, right? You know that if you don't take the test and if you don't do well, then you're going to get a bad grade. But you're willing to sacrifice your time to study, and you're willing to sacrifice opportunity to go out and play soccer or computer games in order that sometime in the distant future, you will get some nice reward that you will find valuable, and it will help you survive in the world. Now, with humans, this can get very abstract. We've disconnected ourselves from the material things in the world where we get pleasure out of ideas. People will work very, very hard in order to make a discovery, even though they may not get any monetary reward for it. So it really is a very powerful way that we have of internalizing the things that are important for us and being able to weigh the different options that we have. Now, there is a relative of octopamine in our brains, and it's called dopamine. And it was known for quite a while by psychologists who studied the part of the brain that the dopamine cells are concentrated in. It's called the ventral tegmental area, or VTA. That they knew that if you stimulated that area in the rat brain, for example, that the rat would be willing to do anything for you, anything. So, for example, suppose that you wired up that electrode that stimulated VTA to a little keypad, and the rat had the option of pressing the keypad or eating some food. What do you think the rat's going to do? The rat sits there and presses that keypad over and over and over again. In fact, it will do that all day and all night, and it will die from starvation before it touches the food. That's how powerful this stuff is, this dopamine, right? And of course, psychologists felt that they really had tapped into something really basic and fundamental. Well, here is that area, the VTA, that I just told you about in the human brain. It's buried deep in the brainstem, down in the bowels of the brain. But its neurons project very widely and diffusely throughout most of the brain, and especially into the prefrontal cortex. This is the front part of the brain that we know is particularly important for making decisions, planning for the future. It also projects to another area of the brain that we're going to see in a little while, called the basal ganglia, and in particular an area called the striatum. We're going to come back to that toward the end of the lecture. But here I just want to show you how these neurons that carry this neuromodulatory substance dopamine, which has a very powerful effect on both behavior and on plasticity of synapses, is really a very, very broadly transmitting system, a very powerful system. And it's an ancient system because of the fact that it's in all vertebrates. It's not just present in our brains, it's present in your dog's brain, in your cat's brain, in your frog's brain, in your fish's brain. It's very, very ancient. So the secret of the dopamine neuron was discovered by Wolfram Schultz, who was studying learning in monkeys. This is Wolfram Schultz and friend. Wolfram is on your right. He's at the University of Cambridge. And when people first started recording from these neurons, they couldn't figure it out because they didn't fire very often. They puttered along at around 1 spike per second. And sometimes they went up a little bit, and sometimes they went down a little bit, but there wasn't very much signal there, and it was really a little bit mysterious. They thought maybe it has something to do with reaching, because they sometimes got a response when it was reaching for something it liked. And finally, Wolfram figured out the secret. So here's the secret. That first of all, when you give a reward to the monkey, and for a monkey that's been, a drop of juice is a wonderful reward, because monkeys love juice. And they have preferences. Some like orange juice, some like grape juice. But in any case, if you give them a task to do, they're very happy to sit there for hours and hours pressing a button or doing a visual discrimination in exchange for a drop of juice. Now here's what's called a rastergram. Each little dot here represents a spike, and they've been put in a straight line on top of each other. There's about 20 trials here in which the monkey has been given a reward just for sitting there and paying attention. So this is without any learning going on at all. And you'll notice that this is 1 second, that's 2 seconds, so about 100 milliseconds after the reward, there's a burst of spikes, and if you average by making a little histogram up here, you simply take all the spikes that occur at a single time and then bin them. You can see that there's a peak of firing. It's about a 1 hertz average here, and then suddenly you get a burst of firing that maybe is 10 hertz. Okay, now let's train the monkey to do something. Let's train the monkey to only, when a light goes on, for example, yellow light goes on, that's when it's going to get a reward. Sometimes when the light's not on, there's no reward, but the monkey at the beginning just doesn't understand that, right? What's a yellow light? So after about a couple of hours of training, this is what that same neuron is doing. The same neuron is now firing to the yellow light. The CS means conditioned stimulus. Here it's a light that has no meaning to the animal before it's been trained. But now look at this. Look at the reward. The neuron is no longer firing in response to the reward. It's as if it's indifferent. It doesn't really care about that reward anymore. This neuron now seems to be firing in response to this yellow light. And yellow light meant nothing before. But the yellow light means a lot to this monkey now, because the yellow light predicts the coming of a reward in the future. That turns out to be the key to unraveling this whole story. It turns out that this neuron doesn't actually, it's not a reward neuron. It doesn't fire at the reward. It fires when it comes across a stimulus in the world that predicts that if you follow up with an action, you're going to get a reward. It's predicting future rewards. And that turns out to be exactly how the temporal difference algorithm works. The temporal difference algorithm predicts that once you've learned the meaning of a stimulus, you basically cancel out. You subtract away what you predicted, what reward you predicted, from the actual reward you got. And if the two are exactly the same, if you got exactly what you expected, then you don't fire at all. That's exactly what's happening. But, okay, that's a nice story, but how do we know that that theory is correct? Well, and here's where Wolfram Schultz had a brilliant idea. He said, maybe there's something actually happening there, but it's masked by the fact that there are two things which are being subtracted. And now I'm going to do something really nasty. Every once in a while, the yellow light comes on and there's no reward, right? He tricks the monkey. Has that ever happened to you? You really get upset. I deserve that. I really, you told me I was going to be able to watch the TV show tonight, and somehow you feel that, you expected it and there's something missing in your life as a consequence. Well, here it is. You take the trials where the reward was predicted but no reward actually occurred, and you'll notice that there's a hole here where there's an absence of spikes. And that reveals the fact that there was something that was in fact present there. The reward was subtracting from some negative input, and you take the reward away and you reveal the negative input. Then that negative input is the predicted reward. So this is a confirmation that in fact temporal differences really are being computed by that neuron. Temporal differences in terms of the difference between the expected reward and what you actually got. So this was a really important observation because of the fact that it revealed a whole new mode for the brain that previously had been neglected or ignored. The fact that, for example, we have percepts, that we see things and objects. It's always been assumed that it's being represented in the brain the same way, regardless of whether you're hungry or you're happy or you're sad, that an object is an object, or a rose is a rose is a rose, right? That's not true. It turns out to be not true for the sensory parts of the cortex that represent visual objects. It's not true for the somatosensory cortex. And here's a very simple experiment that was done by Paul Glimcher, who's at New York University. And I don't have time to go into the details, except that he's recording here from a part of the brain called the parietal cortex, which gets visual inputs, right? So these are visually driven neurons. And the monkey is trained in blocks and is trained to fixate and then at a certain time to move its eye either to the right or the left. And the reward that you get for moving to the right or the left is different, and depending on the block of trials that you're in. In block number 1, you get twice as much reward when you move your eyes to the right than the left. But in block 2, you get twice as much when you move it to the left compared to the right. And this stimulus here is one that you move your eye to, is one that the neuron is firing the neuron. So for example, here is one of these neurons firing at a very high rate. And just forget the histograms, but just look at the curves here, right? These curves are firing rates, and that's around 75 spikes per second since the onset of the target, which is to say the stimulus that comes on at this location or this location. And these are blocked trials, and for the case when the monkey moved to the right, and there were 2 different trials, 2 different blocks. One, again, the same behavior, the same movement in both cases, but what's different is that the monkey knows it's going to get rewarded because it's in this block twice as much for these trials, the red trials, compared to the blue trials. So look at these 2 different conditions, right? But they're both the same neuron recorded under these 2 different conditions. And you'll notice that the peak firing rate here in block 1 was much, much higher, was more than twice as high for the same object and the same movement, right? The only difference is that the probability of reward, the amount of reward, has changed. It's doubled. And the firing rate of the neuron reflects the fact that the value of the stimulus is different. And that means the neuron is coding not just the properties of the stimulus, the abstract sensory qualities, but it's also coding the value that that stimulus has for the monkey, the intrinsic value. And that's true of many, many neurons in the sensory parts of your brain. That we've discovered that your brain looks at an object differently depending on what you think is in it, right? You can have a box. The visual stimulus is identical. If you think there's a reward in it for you, your visual system is going to be much more interested in it. It's going to fire more spikes. And that will guide your behavior. It will bias and make it more likely that you'll be attracted to the box, that you'll want to open it, and that makes a huge difference on your behavior. So that's really a discovery that has happened just within the last 5 years that this has changed. And there are a lot of follow-up experiments that have been done by Bill Newsom, recording from other parts of the monkey brain, showing exactly the same thing under very, very, with different types of tasks, different conditions. There's no doubt now that the neurons are coding, in addition to the features of the object, also the probability of getting reward and the value of the reward. In other words, it might fire more for orange juice if the monkey prefers orange juice compared to grape juice. Now, there's another neurotransmitter, neuromodulator, another system in the brain that uses another substance called serotonin. And serotonin is another one of these ancient neuromodulatory systems, but the function of serotonin is really a mystery. We just don't yet know as much about it as we know about dopamine, for example. But we have a pretty good guess, and I'm going to tell you what that guess is. But first, let me give you a little bit of background on serotonin. So, Measurements have been made on monkey brains to see the level of serotonin activity and try to correlate it with behavior. And here's what they've discovered. These are in vervet monkeys up at UCLA. First of all, the vervet monkeys have a pecking order. There's an alpha male, and then there's a subordinate male, and then that goes all the way down to the most junior member of the troop, monkey troop. So, first of all, if you measure the baseline level of serotonin activity, you discover that the alpha male has the highest serotonin activity. And the guy at the very bottom of the pecking order has the lowest. And their behaviors are very different. Now you might think that the alpha male fights a lot, right? Because he's the tough one. Not so. Turns out the alpha male rarely fights. He only fights when it's really important. He has his lieutenants fight. He uses social interactions to be able to preserve his position in his society. So here's an experiment that was done. What you do is you take the male away, out of the alpha male away from the troop, so that he's no longer there. And now what you do is you give a drug to one of the other males, somewhere toward the bottom, to raise their serotonin levels, right? So that it goes up. What do you think happens? Well, a couple weeks, takes a while. After a couple weeks, their status goes up. And what's interesting is that the males don't notice it. The females notice this first. Females immediately pick up there's something different about this guy, right? Maybe he's gentler, or maybe he's more trustworthy, or whatever it is. Maybe he brings them food. But suddenly the females start paying more attention to him. And the next thing you know, the rest of the males follow. He becomes the alpha male. Do you know what that drug was that they gave that monkey? Prozac. Prozac is a serotonin reuptake inhibitor. So the serotonin is squirted out by the neurons, and then it's taken back up. But if you block that reuptake, it increases the concentration and the activity, that impact that serotonin has. And that's what they did. They were able to manipulate the social behaviors of the monkeys by simply changing the balance of serotonin. It also is correlated with a lot of other types of behavior. Just briefly, if you're interested. So the low serotonin monkeys take risks. For example, they leap between branches. They take big risks. They leap 10, 20 feet, and they often fall. They get into trouble. Monkeys have a very traditional way of dealing with altercations. It doesn't make a lot of sense to fight in order to settle something when it's clear that one of them is bigger than the other. So what you do is you posture, you raise your voice and try to sound as low as possible so that you can sound like you're big. And then after a few minutes, it becomes clear that one of them is going to beat up the other one, so the one becomes submissive and everything's fine. Everybody knows their place, right? Well, these low serotonin monkeys don't wait. Even if they're smaller, they just go for the jugular, and of course they get smashed. So they're risk takers. So it's an interesting, the whole set of behaviors is different. So about a year ago, one of my former postdocs, this is Kenji Doya, who's in Japan, did an interesting experiment. Okay, so he was interested in this temporal difference learning algorithm I told you about. Now, it turns out there is a variable parameter in that model that has to do with what's called the discount factor. So what's the discount factor? Well, the question is, should you be making decisions on the basis of immediate return, right, instant gratification, or should you be making decisions on the basis of long-term gains, right? That's the discount factor. If you think that the discount factor is 1, that the future gain is as good as getting it now, then you are going to be willing to do sacrifices here and now in order to be able to accumulate the gains in the future. But if your discount factor is 0, you don't even care if there's a tomorrow. Basically, you just want that food that right now. You want to get your reward right now. Right? It's an interesting issue. Is different people have different views about where you should be along that continuum. So Kenji did a really clever experiment, and this is now using a technique called functional magnetic resonance imaging. It's a way of looking at brain activity inside the human brain. And here at UCSD, we have a really excellent functional magnetic resonance imaging center with 4 magnets. And so there's a lot of experiments going on there. And this is just an example of what you find when you do one of these experiments. So first of all, this is a cross-section of the human brain, like the one that you saw. This is a real human brain. And in this particular experiment, the subjects were taught, they learned a couple of different tasks to do that involved making decisions. And the different tasks had different degrees of discount factor built in. So that in one case, in order to be able to get the reward, you have to go through many different intermediate positions. You have to make choices that in the short term were negative, that actually punished you. But if you made enough of those decisions, then at the end you get a big reward. So you have to really be willing to do a lot of work in order to get that reward. That was the discount factor of 1. Gamma is equal to 1. That's blue. And then he also had a game where basically as long as you press the right button, you get a reward. This is the very simple task, and that's a discount factor of 0. And then he asked, where in the brain is there activity? And the way this technique works is by measuring blood flow. Where there's more activity, it's thought there is more blood flow. Although the link there has not really been worked out in detail, but nonetheless, that's what we're going to assume. So here's the activity that was measured during these tasks. And there's 3 areas in particular. This is an area in the cortex, in an area called the insula. This is an area in the orbital frontal cortex, and this is an area in the basal ganglia. I told you about this earlier. Now, first of all, in the case where the discount factor was very, very high, right, when you're sacrificing for the future, the Raphe nuclei lit up. The Raphe nucleus is the nucleus containing the serotonin neurons. So that's interesting. So that means that you needed the serotonin neurons to be highly active in order to be able to go through this punishment that you're getting in order to get to your final goal, right? And what's interesting, if you look carefully at these areas, it's been color-coded here according to where the peak of the activity was for different discount factors. You'll notice here, it's most clear here, that there is a topographic map in that region such that the discount factor, the neurons here are active when the discount factor is very high, when you're sacrificing, and when the discount factor is low, when you're learning a task where you get instant gratification, the activity peak is in this region here. This suggests that there are different circuits in different parts of your basal ganglia which are engaged depending on whether you're planning for the future or whether you're just trying to get your feedback, get your reward now, immediately. And it also suggests that serotonin, this mystery substance, might have something to do with this continuum between being willing to wait for a long time before you get your reward compared to someone who needs to have it right now. And that may have to do with both the level of activity and also the different circuits that are being activated. This is all, by the way, very provisional. What I've told you just now may change because of a new discovery. I can't tell you right now much more about it because it's so new. This was published just last year. But it's really an intriguing possibility, and it's a possibility that was developed through a theoretical model. The idea is that by having a theoretical model of what's going on in the brain, we can design experiments, we can go in and ask questions that we didn't even know before of something that might be going on in some part of the brain. So this is I'm now coming to the end of the talk, and this really illustrates the fact that we have to make decisions every day. every day we're making decisions. Should I come to this lecture? Well, the title is kind of interesting. Who knows? Maybe it'll be fun. On the other hand, I might fall asleep. Or should I go onto the internet? Maybe I can get more information by surfing the web, right? How do we make these decisions? Where do they come from? How do we weigh apples and oranges? I think it's because of dopamine. I think that the dopamine system is really the lingua franca of how we weigh differences. We imagine going to and doing something, and then our dopamine neurons tells us. It's a gut feeling we've got. This is going to be fun, or maybe this is not so much fun. Now, what's really fascinating is the fact that all addictive drugs work through the dopamine system, either by increasing dopamine or by blocking reuptake of dopamine. Cocaine, for example, is an addictive drug. Heroin, all of them work through that same, we think, neuromodulatory pathway. And what we think is happening is that these addictive drugs, just like the self-stimulating rat, are hijacking the brain's internal predictor of future reward. And if you're addicted, you are willing to do anything to get that reward, because that's the brain's ultimate substance for telling you what's important. And if you're addicted, it takes over your will. You will do self-destructive things. You will do bad things that will alienate you from your loved ones. You'll do bad things that will get you in trouble with the law. This is really bad news. It's really you're tampering with the brain's really basic circuitry here. That's really a very fundamental part of how we're motivated and what determines our decisions and our actions. And so finally, the last point I want to make has to do with the importance of timing. Because remember, going back to the bee, I was telling you about how the bee gets reward. So it turns out that none of this system works if you get the reward before the stimulus. There's an asymmetry. There's a causality that's embedded in the actual learning algorithm. If you get the reward and then you get the stimulus, you don't learn anything about the stimulus. And there's a very simple reason, is that it's unlikely that the stimulus had anything to do with getting the reward. It was some other cause. Something else was responsible. And we have built into our very nervous system the very essence of causality, that you only are going to learn things that happen in the right sequence. It's also important, obviously, if you're in an uncertain world, to be able to act very rapidly on a very short time scale. To be able to get the reward, you have to be in the right place at the right time and be sure to have your mouth open. Thank you.